Hello, my name is Macey Rains, and this is my research project. Please forgive me of any mispronunciation of any of the words because I'm not a scientist. But the title of my project is Thiamethoxam and Clothianidin negatively contribute to queen bee reproduction by increasing ovary size. So let's start off with a couple definitions. So Apis mellifera is a western honeybee. Then the Thiamethoxam and Clothianidin are neocotinoids. So these are different pesticides. And then specifically, we're going to be looking at a queen bee. So that means a honeybee that is fully developed. That's a female, and she's a mother of the hive. So, why are we studying queen bees? So, bees are vital to global biodiversity and food security through their pollination of plants, including several key crops. Bullet point two is that there's an overwhelming amount of evidence now suggesting that numerous wild and managed bee populations are in decline. So, this is likely due to pressures like these pesticides. Also, it could be due to climate change and changing land use. Um, then the third bullet point is that there's concerns over the human food security and maintenance of biodiversity because this neocotinoid class of chemical pesticides has recently received considerable attention. And I apologize, it's Apis mellifera. Anyways, moving on to the research question. So, how do thiamethoxam and clothianidin affect Apis mellifera ovaries in queen bees? So, the method done, this is talking about the experiment aspect. So, queen bees were allowed to sexually mature, and surviving queen bees were sacrificed to examine their reproductive systems. They used like a group study of bees, and this study followed the statistical guidelines for the Apis mellifera research. The amount of pesticides used was 4 ppb and 1 ppb of each of these here. And then each colony received 100 grams of pollen supplement every day for 36 days to ensure that colonies contained young bees exposed to the neocotinoids during queen rearing. Supplements were well received but never completely consumed during each feeding period. So it would be interesting to go back and have them do this experiment with the bees fully taking in the amounts. So they created this like nucleus. This is what the outside of it looked like, but it just acted as the place that the bees lived until they were ready to take them out for dissection. So the surviving bees were removed from their colonies, and they used carbon dioxide to put the bees to sleep to allow for the inspection of their reproductive anatomy. So they used some other scholarly research to make sure that they had proficiency in their dissection techniques, just because this was like a more complex procedure to study the reproductive anatomy. So some of the specific tools that were used were using the buffer 32 to study the sperm. They also stored sperm using a hemacomometer, cytometer, and they also used compound microscopy. They also were able to check the viability of the sperm using a laboratory kit listed here and suspension solution. And then lastly, they could see the number of ovaries by using real-time counting under a microscope and a fine needle. So that's there. Here are the figures. So the one on the far left shows the queen survival after 4 weeks. There was like no significance between the amount that survived and didn't survive of the ones that there's like no significance between treatments of the control and then the ones treated with the neocotinoids. But the ones that did survive following on the second figure, part A, you can see the percent of honeybee queens that laid worker bees, and then part B is showing the percent of honeybees that were alive and had produced diploid offspring. These both had significant differences between the two. More results included that queens surviving 4-week observation periods, ovary sizes of those exposed to the neocotinoids were 6.8% larger than those compared to the control. Neocotinoid queens had fewer percent stored sperm and a 9% lower proportion of living versus dead sperm. Significant effects of neocotinoids on honeybee queen anatomy and physiology were just shown here in these 3 different charts. And then increased ovary size suggests that neocotinoids can affect development of queen reproductive systems. So for conclusion, Thiamethoxam and Clothianidin negatively contribute to the queen bee reproduction by increasing the size of ovaries. Exposure to neocotinoids significantly impacts honeybee queen anatomy and physiology, as evidenced by larger ovary sizes, reproduced sperm storage, and decreased sperm viability. These findings suggest that neocotinoids may disturb the development and functionality of the queen's reproductive system, potentially compromising colony health and sustainability. So the significance of this is that the information is important because insect pollination services are particularly important for safeguarding agricultural productivity and ecosystem sustainability. The queen specifically is really important to study and understand because she is like the central survival and productor of the honeybee colony. So she's solely responsible for reproduction, and anything that disrupts her reproductive system, such as reduced sperm storage and viability, the increasing of her ovary sizes can weaken the colonies or have a potential colony collapse. Moreover, larger ovary sizes, while seemingly a positive adaptation, may indicate developmental stress or maladaptation due to neocotinoid exposure. This highlights the broader ecological risks of neocotinoids, emphasizing the need to evaluate their use to protect pollinators' crucial role in the global food production and biodiversity. And this is my references, and thank you for listening.